All right, thank you so much, Andrea, for the very generous introduction. And of course, thank you all for being here and hosting me. And I would like to say that, I mean, feel free to kind of stop me and ask questions. It's what, past 9, and I just want to make sure that I don't doze off time to time, so keep me awake. So and yeah, I have to say it was an amazing thing to actually meet up with Andrea when I got to Singapore, and the best discussions I've had generally in science and just about everything else. It's been quite a journey actually since then, and. Yeah, and I've always been super impressed by the imaging that you guys do, and that's something that we are sorely lacking here, but we do a little bit of the genomics that I'll talk to you about. So okay, so without further ado, let me just start. So tumor evolution through the looking glass of single cells. You can see my slides, right? Yeah.
Yeah. Perfectly.
So we're very interested.
Sorry, Ram. I would recommend everybody to just mute while Ram speaks, and then if you need to say something, just raise your hand.
Yeah. So we look at cancer through an evolutionary lens because we think cancer evolution, tumor evolution in general, is very Darwinian. Mainly because of which I'm sure of a remarkable amount of inter-patient as well as inter-tumor heterogeneity, and the fact that tumors are fairly dynamic in nature. Can you see my pointer? Let me just change it to a laser pointer. Yeah.
Yep.
So, so we know that there are different clones and subclones of cells that show different selective fitnesses, just like a diverse population undergoing evolution under different selection pressures. And of course, tumors are highly plastic and dynamic, that they change over time, so they can gain mutations, they can alter their epigenome, and thereby resulting in altered transcriptome, which then helps them survive a selection pressure. And that selection pressure could be of a drug or, say, a metastatic environment, the typical seed versus soil hypothesis, how a tumor cells, even during metastasis, can survive or undergo immune evasion from the body's own immune system and eventually metastasize to a different site and then colonize and then grow. So fundamentally, what I'm really interested in, and my lab's interested in, is really understanding this tumor heterogeneity, and then how heterogeneity drives evolution, eventually resulting in treatment failure. And from a clinical standpoint, of course, if we understand this better, then the major question that we want to ask is, how do we improve clinical outcome from discoveries made in this fundamental biology of tumor evolution? So, in terms of what we've been doing the last several years now, is really trying to map these trajectories of evolution really at the level of single cells. And we are interested both in tumorigenesis, so how simply a normal cell becomes tumorigenic, becomes transformed, and how it interacts with the tumor microenvironment, which may be helping in the entire tumorigenesis process, and something that we like to call it the tumor evolution of the past, so what's happened before. Of course, vast majority of patients actually succumb to cancer mainly because of tumor progression, and this is kind of the future tumor progression under the selection pressure of drugs, or they metastasize. So, I mean, most cancers, of course, are not lethal in terms of the primary tumor, but most patients die of tumor progression when it becomes malignant or resistant to therapy. And that's something that we would want to understand at the single cell resolution. And why we want to understand at the single cell resolution is really we want to identify these founder clones or subclones that then are either selected or they adapt to different selection pressure to give rise to a resistant or metastatic phenotype. So long-term goal and why we want to do it is the idea behind this that, well, if we identify these earliest events and molecular mechanisms of trajectories of tumor evolution, then perhaps we can intervene early and therefore prevent or delay the evolution of resistant or metastatic disease. And lending this credence to this idea that, you know, of evolutionary therapeutics, that is perhaps tumor evolution, or the processes that drive tumor evolution can be used as therapeutic target, and which then results in, you know, in the long-term goal that, you know, if we can always stay ahead of the cancer by predicting any given cancer's next evolutionary move, then we can intervene early and therefore convert it into a chronically managed disease rather than having to succumb to the disease. All right, so that's really the general outline. The kind of approaches that we take, well, we think the best model to understand cancer is really the patient itself in the clinic. So of course, we do a lot of longitudinal studies for mapping trajectories of evolution when you go from, you know, pre-post drug treatment or primary and metastatic tumors. So that is basically patients in the clinic, but we, you know, not for all the different cancer types, it's not necessarily easy to kind of always get the clinical samples because of the regulatory concerns. So in those cases, we basically use patient-derived models. And of course, I'm sure you know this, patient-derived models can come in a variety of flavors: patient-derived organoids, primary cell lines, and patient-derived xenografts. And these, of course, can be then used to model cancer progression to resistant to metastatic disease. They can be extensively profiled genetically, functionally, using, you know, drug screens or CRISPR-based screens. And then, of course, these discoveries can then be clinically validated, which then hopefully will contribute towards clinical applications of predictive biomarkers and identifying new therapeutic targets as well as drug candidates. And some of this work, I'm not going to go into it, but there are a few papers that we published around 2017 and 18. If you're interested, then have a look at these papers, which basically show how we can use these patient-derived models to identify new prognostic biomarkers as well as therapeutic strategies based on patient stratification.
Sorry, Raman.
Yeah.
Just to break up the ice a little bit, I would like to ask you, in your experience, which one of the patient-derived models is the most stable, I mean, genetically over time?
Yeah, that's a good question. So we are writing up another paper actually based on just that. In fact, from what we've looked at in head and neck cancers and colorectal cancers, whether we look at organoids or with primary cell lines, we do not see much of a genetic drift. In fact, they look somewhat, in fact, much better in terms of retaining the molecular subtypes, which is based on consensus molecular subtypes, the CMS subtypes, based on RNA-seq, as well as the genetic signatures. If anything, we see more of a drift in the PDX models, where you see a typical selection or very strong selection sometimes, or mouse-specific selection. So we think that if you can, in fact, grow these tumors directly into organoids or cell lines, they seem to be fairly stable, at least over 10 to 15 passages. So everything that we do is usually on fairly early passages. But what the study will show, hopefully, is that in fact the consensus molecular subtypes are identical. You know, so much of the signatures is retained, and it's a question for discussion, like why is it retained, for example. So we do think, you know, the cells remember their surroundings, and there's some kind of an epigenetic memory that keeps the signature at least for some time, at least over several passages, and they don't necessarily go away. And in terms of genetic mutations, yeah, they're fairly retained. Of course, it won't be identical, but, you know, R-square values of more than 0.85 or 0.9, so it's pretty decent, yeah.
And also the variability stays stable.
Pardon me?
The variability, the heterogeneity of the populations remains stable.
Yes, so
Okay,
heterogeneity of the populations also remains stable with single cells, yeah. And Hans Clevers has shown that as well, at least in colorectal cancers. So why do we want to do, you know, why are we interested in looking at this at the level of single cells? And in some ways, it's really to parse out all the different nuts and bolts that, you know, make a functional cell. In this case, you know, an example of a car. So, you know, with single cells, well, you can identify all the individual parts, basically, which are the different cell types or cell states. And then you can localize them in time and space. So where does the wheel go, and where does the steering wheel go, where the seats go, etc. And that's basically basically a lot of effort globally that's being done in terms of human cell atlas. So that's basically simply helps you generate an atlas of all the cell states or cell types that you might find, say in a tumor or even in normal tissues, right, and organs. However, what's really important is to not just have this atlas, which basically tells you all the different kinds of cells that are there, but what what the atlas doesn't necessarily tell you is that how they work in time and space. And for that, you have to kind of go a step beyond atlasing and try to understand, well, what are the cell-cell interactions? What are the cell signaling nodes? What are the networks that are driving that are making this engine per se, and this is of course how similar to maybe how a cell would metastasize or how it interacts with the microenvironment, with the immune cells, with the stromal cells. And that's where I think a lot of novel biology is left to be discovered. And of course, if we can figure that out, then we can identify how all of these modules work together basically to make a functional cell, or in this case, drive the car. So in terms of just very briefly, what we've been mainly working on is really kind of the tumor cell intrinsic mechanisms of tumor evolution. And I'm not going to go into the details, but these are some of the papers where we've used patient-derived models, in this case in oral squamous cell carcinomas, and we identified a SNP in the long non-coding RNA, which is highly predictive of TKI response. And we also identified these rare clones of YAP high cells. So this is a key transducer of the hippo-YAP signaling pathway that then drives the metastasis as well as resistance against tyrosine kinase inhibitors. We also had another paper recently on drug-induced tumor evolution, in which we could show that cisplatin treatment would basically induce a stem cell switch. Details are not really important, but pioneer factors from SOX2 to SOX9, and this stem cell switch results in an epigenetic rewiring of the cancer cells to activate these resistance and metastatic mechanisms, and you can reverse that basically using co-treatment with epigenetic drugs. So similarly, we are studying these tumor cell intrinsic mechanisms of evolution in terms of metastatic colonization, and we have some really interesting data which I won't have time to talk about in terms of fatty acid metabolism and how it could be driving liver metastasis in colorectal cancers. And another story that hopefully you'll see soon, that's just been submitted, where we can use single cell transcriptomics to identify clonal heterogeneity and as well as clonal heterogeneity of drug response. So based on the heterogeneous population, we can come up with specific combination of drugs that kills the tumor, that targets the tumor as a whole, rather than the specific population. So really an additive effects of clone-specific therapeutic responses that then could be used in the clinic. But what I'll focus today's talk really is about the tumor microenvironment and how do tumors thrive in the context of this tumor microenvironment. And just like in this, a beautiful coral reef ecosystem, where there's a lot of mutually beneficial coexistence, right? A lot of symbiotic relationships, and if you kill off one species, and then often these corals would die. It's the same thing with the tumor, and I think it's becoming increasingly clear that to really understand tumorigenesis or tumor progression, we really need to understand how the tumors evolve in the context of its ecosystem. And in this case, of course, you could see that it's not just the tumor cells, but of course all the neoangiogenesis or vascular genesis or endothelial cells that provide the nutrients through blood. And of course you have cancer associated fibroblasts and stroma, ECM remodeling, and of course the immune system that could be either favorable for tumor growth or could be unfavorable. And I'm going to get into more of the details. And so much of the work in the last 3 years or so has been focused also on tumor extrinsic mechanisms or tumor cell extrinsic mechanisms that are driving tumor evolution, with a specific focus on tumor microenvironment. And we have several stories that are building, but I'm going to mainly focus on this bottom left, which is on looking at how the tumor grows and survives in the context of its ecosystem in human hepatocellular carcinomas. And this is partly funded by the Human Cell Atlas. Sorry, is there a question? So, so partly funded by the Human Cell Atlas, as well as a large collaborative grant, LCG as it's called in the Singapore system. So it's like a PO1 grant in the US. So it involves several clinicians, bioinformaticians, and of course, cancer biologists like my lab. So let me get to the story. So this is going to be the focus of the rest of the talk, is really tracing this tumor heterogeneity and co-evolution of not just the malignant cells, but also that of the microenvironment during tumorigenesis. And of course, we're very interested in what happens under the selection pressure of relapse, and that's going to be a separate story, not for today, but I'm mainly going to talk about tumorigenesis per se. So before I talk about it, let me actually acknowledge the people who've been driving it. So one of my most amazing postdoc, Ankur, he's been leading the single cell effort, and especially the work on single cell genomics and transcriptomics. He's recently graduated from being a postdoc to a colleague. He's taking up his own position at the Curtin University in Australia in Perth. And he'll be starting his own lab in January. Justine is an ace bioinformatician who's been doing all the work with single cells. Ria was a key member of this project in terms of all the single cell, the isolation and the process of using single cell transcriptomics. Piers Chow is a surgeon at the National Cancer Center, Singapore, who provided us with all the liver biopsies and the resected samples. And we work very closely with Florence Genot, who's one of the gods of myeloid lineages, or, it looks at ontogenies of monocytes and macrophages. And so much of the cancer immunology in terms of myeloid biology, we've basically learned from him and collaborated with him on this. So the work basically started based on this a huge collaborative network that Piers has set up in the area of liver cancer, and it's called the PLANET study, which is Precision Medicine and Liver Cancer Asia Pacific Network. It's a mouthful, but basically in the APAC region, so it's Malaysia, Vietnam, Cambodia, of course, Singapore, Indonesia, and Australia as well, and some members in Japan and Korea. So it's a multi-center site for clinical trials or translational clinical trials. And one of the main goals for this PLANET study was really to kind of dissect heterogeneity, and not just in terms of genomics really, but we're very interested in epigenomics, heterogeneity in transcriptomics, as well as in this case, immunomics and metabolomics as well. You can imagine the clinical protocols that have led to the creation of this and made this happen. But what's really made this possible is that a specific clinical protocol which allows us to get tumor adjacent normal, and then we cut these tumors up into multiple different pieces. So you can get the tumor adjacent normal, you can get the tumor periphery, and kind of the intermediate region in between the periphery and the core, right? So for every patient that comes in, we try to get at least 3 to 4 sectors basically. So it's multi-sector. And then all those sectors go towards sequencings for DNA, RNA, epigenome, and of course some go into flow, FACS for CyTOF, for immunology, as well as metabolomics, right? So this is how this work really started.
Sorry, Rama.
Yeah.
I have a question. So when you make this section, this is only topologically defined, or you do look for some change in
this is based on
large scale?
This is based on what the pathology tells us. Yeah. So there is a pathologist in the operation theater working with the surgeon, and when the tumor comes out, so all of this needs to happen like very, and the workflow needs to be really fast. And so they define what the tumor border is, and then it's about the tumor normal. Okay, I have to say, well, tumor adjacent normal is never quite normal, at least in terms of the immune cells, right? There's a lot of inflammation, and these are generally cirrhotic livers, which have now become HCC. So there's, fibrosis, etc. But it's about 2.5 to 3 centimeters away from a well-defined border. And then from the tumor that is resected, then they cut it up into different pieces. And, of course, some goes for further pathology and diagnostics for the clinical samples, and rest goes towards research. Yeah. So purely based on pathology and morphology. Yeah. Nothing else. Okay. So, so the first thing initially, well, as part of Human Cell Atlas as well, we basically just wanted to build a comprehensive single cell atlas, and then hoping that we'll learn something from the single cell atlas about the biology of HCC. So we followed the typical 10x genomics pipeline. So again, this is the tumor sector, and you have multi-sector. So you have one normal and multiple sectors, and this is, this is basically how it looks like. And we did it for about 14 patients. So total, I think about 62 sectors or something like that. And then they were followed up with, of course, single cell RNA-seq. And then I'll tell you more about all the other validation studies that we did with IHC and spatial transcriptomics and FACS, all right? But what we do is that as soon as the tumor comes in, we dissociate it, and there's a dead cell removal process. And after that, we do a sort for anti-CD45. So basically a positive and a negative sort. CD45 enriches for immune cells, and negative enriches for the tumor cells. And then we do a one-to-one mixing, and then we put it through the 10x Chromium platform. So encapsulate them in these gel bead emulsions, and and just follow the 10x protocol, which is then eventually sequenced, right? So I'm not going to go into details. I'm sure many of you know this. The only thing I would stress upon over here, the reason we do this sorting and then mixing again is really because hepatocytes actually don't like dissociation so much. So we were losing a lot of hepatocytes. So in fact, there weren't many liver cells per se in HCC, which is why we had to do this enrichment protocol. And now we have enough liver cells, enough hepatocytes in the single cell protocol. All right, so how does it look like, right? So this is the multi-sector atlas. So 14 individuals, like I said, 62 sectors. So, from some patients you get 2, other patients you get 3 or 4. So that's basically a sum of all the sectors. You're looking at about a total of 80,000 good quality cells. So this is after all the quality control and everything has been done. And I'm sure many of you have looked at these U maps or T-SNE maps. Basically, it's clustering of cells. Each dot represents one cell, and they cluster based on transcriptomic similarities. So cells within the same cluster are basically the similar cell types, right? And over here, what you see is that we identified about 29 different cell types, so 0 to 28, so about 29 different cell types in HCC or in the liver. And on the right-hand side, we've colored the same clusters based on the patient identities. And you can see most of these clusters are confettis, so suggesting that there aren't any patient-specific batch effects. Only the batch effects that you see are in these clusters, and hopefully that's very clear. So these are all single colors. And as it turns out, these single colors are the ones that are in fact hepatocytes. And that's something you'll see it in just about every single cell tumor atlas, because there are very patient-specific transcriptomes which are based on patient-specific genetic signatures and mutational signatures. And of course, this is only from 14 individuals, of which we got hepatocytes, good hepatocytes, only from about 6 individuals. So I think if you go into now hundreds of individuals at some point in time in the future, then maybe you will start seeing similarities. But what's, this is very commonly observed. The other thing, the only confetti that you see or rainbow that you see is this small cluster, which is also all hepatocytes. And this is coming from every patient, and these are all normal. And these are progenitor-like cells. So they are progenitor cholangiocytes and hepatocyte population with expressing keratin 8 and 19. This is just to show you how the different atlases look like. So now this is colored by spatial. by spatial coordinates. So blue is adjacent normal, yellow is a tumor periphery, red is a tumor core. And so this is the tumor atlas, this is the liver atlas, right? And because liver is also driven, or much of the HCC is driven by hepatitis B, amongst our patients, we had 6 patients that were Hep B positive and others that were Hep B negative. But, you know, overall, from this very kind of, 30,000 feet view, what you see is that you don't really see a huge difference in these different clusters for Hep B positive and negative. So clusters don't necessarily segregate out based on Hep B expression, apart from, of course, what you would expect from the hepatocytes, right? So clearly some patients who are Hep B positive, others that are Hep B negative, which is there in the red. So that's just atlasing. So after atlasing, what you do is annotation. And again, without going into too much details, you just basically look at the top differentially expressed genes in each cluster with respect to all the other clusters. And based on those differentially expressed genes and a lot of literature reading as well, now there are a lot of single cell cluster annotation or cell annotation pipelines using just bioinformatic tools, but you still have to go back into the literature and confirm that they are indeed the cell types that they say it is. And then you can start annotating these clusters. So again, these all turn out to be hepatocytes, so they all express albumin, for example. These are fibroblasts, these are endothelial cells. This little red cluster is that bipotent, progenitor-like cells that comes from every patient. This large cluster of cells over here are lymphocytes, right? Including CD4, CD8, as well as natural killer cells. These are the myeloid cells, so monocytes, macrophages. This is a B cell cluster. And that's about it, right? And just so that, you get a glimpse of how we go about doing it. So, these are basically just looking at the expression of specific genes, cluster specific or cell annotation specific genes on the UMAP. So you have these endothelial markers such as PECAM. This is alpha SMA, ACT2 and fibroblasts, Catin-19 in the progenitor cells. Hepatocytes are expressing albumin, IL-7R, as well as CD8. So these are the CD8 cells, CD4s. You have the NK cells, MZB1 and CD19, basically in B cells, and then CD14 are the myeloid cells, right? And so again, and this is basically a dot plot showing the expression of these top differentially expressed genes that are specifying certain cell types or cell states, right? And I'm sure you're used to seeing this in single cell data. So let's get on to what we found that was interesting. So, well, the first thing beyond the atlas, and this is where now we're getting into a little bit more of the biology. First thing that was very curious, again, because we had the adjacent normal and the periphery and the core, right? First thing that we were curious to see, that are there any specific clusters that are coming in only, that are found only in the tumor and are different from what's there in the normal. And you can see that the myeloid clusters, for example, right? There are 2 major clusters. And remember, this is again a 30,000 feet, right, view. So of course, cells that are all the myeloids will cluster together, and I'll show you later that you can individually just take this one cluster and then subcluster it into many other clusters. But from this 30,000 feet view, you have 2 major clusters of the myeloid. And what you see is the top cluster is mainly coming from the tumor, but the bottom cluster is mainly coming from the normal, okay, in yellow. Same thing, it's quite fascinating. You find the same very distinct clusters that are coming in endothelial cells, either from the normal or from the tumor. And of course, in the lymphoids, of course, you have this mix. So many of these clusters on the left-hand side, mainly coming from the tumor, and many of the clusters on the periphery on the right-hand side over there and the bottom, in the yellow are mainly in the normal, and the right-hand side mainly coming from the tumor, all right? What would be really interesting, even just over here, that you could see that majority of these cells, in fact, as it turns out, are actually NK and CD8 cells. So much of these yellow cells that are coming from the normal is where the CD8, the cytotoxic cells are, and the natural killer cells are, and much of these peripheral clusters in this blue, yellow, and green over here that is coming from the tumor turn out to be CD4 helper cells with the Th17-like phenotype. I'm not going to go into too much details, but as well as Tregs, suggesting that already that, maybe what we are seeing is that, the tumors appear to be immune excluded, at least for cytotoxic T cells, right? And so understand that better, what we tried to do is that we took these individual clusters and took them, separated them from this integrated data, and then reclustered them to understand this better, all right? And this is what we basically, it's similar to kind of, this is your 30,000 view of Singapore. We are here in this yellow spot where Biopolis is. Of course, in this case, you could see these major clusters, and this is, west and north and east and south. But of course, if you zoom in and start clustering, and this is where I work, in fact, this particular building over here, that's Genome Institute of Singapore. You can start annotating individual cell types and cell states within a given cell type. So you can, start segregating or subclustering the T cells, subclustering the myeloids, subclustering the endothelials and hepatocytes or fibroblasts. What it also allows you to do is start understanding what are these interactions that you see, right? Because once you start doing subclustering, you also get new insights into how these individual cell states or cell types within subclusters may be interacting with each other to perform certain biological processes. So the first thing, like I said, we looked at a subclustering of the lymphoids and mainly the T cells that we looked at over here, all right? And again, now that we subclustered these lymphoids, so I'll just quickly go back, over here we see maybe 6 or 7 different clusters. Now, if you take that out and then you recluster them, what you find is, well, they're about like 25 different clusters. However, they are broadly can be classified into kind of 10 cell types with 25 different cell states, okay? And again, when you look at where these clusters are coming from, so much of these clusters that are marked in yellow are the normal. So from here on, everything in yellow is always normal, and anything in red or purple is tumor. This purple is mainly coming from periphery, red is coming from tumor core. And again, like I mentioned, that we found it in the macro clusters as well, that the vast majority of the cells, that are natural killer cells or cytotoxic CD8 positive cells, they turn out to be mainly present in the normal, in the tumor adjacent normal. Not that it's not there in the tumor, but much fewer basically. And most of the tumor appears to be full of CD4s as well as regulatory T cells that are generally immunosuppressive, right? So what we find in this case, or what this data suggested, that HCCs appear to be exhibiting these features of immune exclusion phenotypes, right? And they're infiltrated with regulatory T cells and somehow excluding these CD8s, NKs, and NKTs. So the question was, of course, why, right? So, well, why is this happening? We started looking at what the reasons could be. And some of the insights basically came from looking at the subclusters of the other cell populations, right? So what you're looking at here now is subclustering of the myeloid population, right? And what you see here again in the myeloid population, so if you remember in the macro clusters, there were really 2 clusters. One was in the north, one was in the south, right? Top and bottom. And so now we see that the top one is now on the right-hand side. The bottom one, the major cluster is on the left-hand side over here. And you can see that, the right-hand, the major macro clusters over here on the right is mainly present or coming from the normal, tumor adjacent normal, as well as tumor periphery. But most of these clusters or subclusters of the tumor associated macrophages, the TAM populations, are coming from the core of the tumor. All right. And of course, on the right-hand side, you have PDCs, you have DC1, DC2s, you have monocytes, you have this blue cluster, which is also more like a transition monocytes. So all of that makes sense. But what was really interesting is that we seem to have identified or stumbled upon a new tumor-associated macrophage population that we, at this point, called the TAM1 population. So if you look on the left-hand side over here. So, what we find is that, there are these two clusters, and the differential gene expression amongst these two clusters is almost negligible apart from a very few genes. So we think they are very similar to each other. We call them TAM1, the cluster 1 and 7. This cluster 2 is, the cluster 3, sorry, is TAM2, and this cluster 5 is TAM3. And these are the specific markers that we find. So the TAM2 population have high levels of expression of SPP1. The TAM3 population has high levels of expression of MT, it's a metallothionein MT1G. But this is the cluster that I want you to focus on, really the TAM1 cluster that has not been described earlier before, mainly because of the expression of this particular gene called FOLR2. And they're also highly expressing high levels of CD163 and CD206. And these generally are associated with highly immunosuppressive macrophages, more like the M2-like phenotype, even though immunologists, at this, from what I understand, talking to them, they don't really think that there is a natural M1, M2 phenotype in vivo, but these are more M2-like, so more tumor promoting, more invasive, more ECM remodeling kind of macrophages. FOLR2 is a folate receptor, by the way. So this was interesting. So, okay, so we find this, but then what about it, right? What is it doing, right? And so before I get into what it's doing, we also, of course, looked at flow-based assays just to reconfirm that indeed there are these monocytes, and then TAM2 and 3 and TAM1 populations. And the TAM1 populations basically has the highest levels of CD206 and 163, as well as, like I said, the FOLR2. So the question is that, well, what's the function of these TAM1 populations, right? And one way of looking at, when you look at single cell RNA-seq data, one way of getting towards function, and this is of course within inverted quotes because you can really infer functions. This is all patient-derived, directly a destructive process, right? So just samples that we get from the patients is you run certain analytical pipelines, and one of the analytical pipelines that's worked out quite well, and many single cell papers now have this, is called cell phone DB, and it's a way. It's a database that is used or a way to infer cell-cell interactions. And the databases are based on secreted and membrane proteins and protein complexes, and then literature-based protein-protein interactions. So what it really looks at is it looks at cells that are expressing specific ligands and other cells that are expressing specific receptors, and then it'll infer interactions based on the presence of these ligand expressing cells versus receptor expressing cells more than just by pure chance, right? And this was developed in Sarah Tackman's lab at the Wellcome Sanger by Rosaire Venter-Tormo, and she has some amazing papers actually based on the cell phone DB. So what you can run then is this algorithm and try to look at what these subpopulations are maybe interacting with in the tumor microenvironment. So what you're looking at on the left-hand side is basically a readout of these cell phone DB based interactions. And there's a lot of kind of, jargon and different ways of showing this, but I'll just walk you through it. So what you're looking at is really the intensity of the inferred interactions, and as well as the size basically looks at the number of cells that could be interacting this way, right? And on the bottom, what you see is, so you have these CD4 interactions with all the different other immune cell types, including CD8, NKs, DCs, and macrophages. And then you have CD8s interacting again with the same set of cell lines, or cell types, sorry, the NK cells, the DCs, and the macrophages. And what you really want to look at, and another thing that you need to keep in mind, is that every alternate row or every alternate column, every alternate column is basically the interaction seen in normal and tumor, all right? So it goes normal, tumor, normal, tumor, normal, tumor, like that. And what I would like you to focus on, and hopefully this is visible to you, let me just quickly annotate if possible, this kind of regions, all right? And what you see here, where did my mouse go now? Eek. Sorry about that. Okay, there you go. I should not annotate
I think it's back. I see
Okay,
moving.
all right. So what you see is you see there's a small dot and a larger dot, and a small dot, larger dot, small, large, small dot, right? So what you see is that in the normal, there is less interaction. With the tumor, there are greater interaction between macrophages and CD4, CD8, NK, and DCs in this case, right? So if you look at all cells interacting with all the others, right? So this is an entire connectivity map in the normal versus tumor. One thing that you could see is that there is a marked increase in the interactions, again, inferred interactions between the macrophages and T cells, right? Goes from about 11 to 22 interactions. And now if you zoom in and try to look at what these interactions are, as it turns out, majority of these interactions between the tumor-associated macrophage populations, whether it's CD4, CD8, or NK cells, turn out to be these immunosuppressive macrophages, right? However, like I told you, that there are multiple TAM populations. There are this TAM1 population, and then the TAM2 and TAM3. I'm not showing a TAM3 here, but basically what we find is that most of the TAM1 interactions or TAM1s are appear to be or are inferred to be interacting much more with the regulatory T cells compared to the TAM2. Now TAM2, which are the SPP1 TAM populations, has been shown in lung cancers as well as melanomas, and I think in pancreatic cancers as well, to be immunosuppressive. But in HCC, it seems that the SPP1 TAM2 like cells, they have reduced interactions with the Tregs compared to the TAM1 population, which is a FOLA2 positive, CD163 and CD206 positive population that we find in HCC. So overall, what it suggests is that this TAM1 population, together perhaps with TAM2 as well, it appears to be mediating these immunosuppressive interactions with cytotoxic T cells as well as helper T cells, but they also exhibit enhanced interactions with the immunosuppressive regulatory Tregs, which are the FOXP3 positive cells, all right? So, so maybe that's what their function is. And you know, but of course we have to show it in multiple different ways. And one thing that we could show with simply spatial technologies, and this is just RNA scope, so just looking at RNA fish. And what we find is that you do find this co-localization of the TAM1 population with Tregs. So this is adjacent normal, and the tumor, there are these large areas of multiple TAMs, which are the CD163 in red. They also happen to be FOLA2 positive, so the TAM1 populations in very close proximity to these islands or clusters of TIGIT and BATF, which I'm not showing you here, but TIGIT positive cells, which are again the markers for regulatory T cells, all right? So indeed, it seems that these TAM1 populations that we find in HCCs could be creating an immunosuppressive microenvironment by recruiting regulatory T cells and by interacting using immunosuppressive mechanisms, so CTLA4, CD86, and PD1, PDL1, for example, and most of the interactions that we see with CD8s or cytotoxic T cells appear to be immunosuppressive. And maybe that's the reason you find less of the NKs and the cytotoxic T cells in the tumor microenvironment, and you find more of them in the adjacent normal, and within the tumor you find more of the Tregs and CD4s, right? So the next question that we asked is that, well, where are these TAMs coming from, right? Where were they? Were they reprogrammed, or is it an enrichment of a pre-existing population? And one thing that you, again, can use single cell data for is something called RNA velocity. So the specific algorithm is called Cyto or VeloCyto. It's from Stan Linnison and Peter Karchenko's group, and this was published earlier, about a couple of years ago, in Nature. And what it does is that you can use this algorithm to actually map trajectories of cells over time, right? And then what it does is that basically it looks at the ratio of newly transcribed RNA versus RNAs that were transcribed earlier. So the whole idea is that if you have a new cell, all right, if you say getting reprogramming of a cell, or there's a cell fate change, which is then making a particular RNA, then in those newly forming or newly transcribing cells, you'll have a lot more transcripts that have not been spliced yet. So what it looks at is a ratio of these unspliced RNA with that of spliced RNA. So unspliced RNA would be more present or present more in a new cell or newly forming cell, whereas the old cell from where it's coming from will have more of the spliced RNA, right? So these, when you look at the velocity graphs, again, projected onto these UMAPs, I'm sorry, this is coming up as very low resolution, but hopefully you could see that what we find is that, you know, when you look at this velocity, so again, most of these clusters on the right are the monocytes and DCs, but many of these TAMs, right, on this TAM1 population. So many of these TAMs, the TAM2 and TAM3, are really coming from this cluster 2 to cluster 0, and then they come in. So it's the monocytes that appear to be recruited, and then they become, when they come into the tumor microenvironment, they're becoming TAM2 and 3. But we were specifically interested in this TAM1 population. And like I said, there are two different clusters, cluster 1 and cluster 7. And when you look at these arrows, it seems that there are two different ontogenies, right? So there are some that are tissue resident TAM1-like cells, so they were already there and they somehow got enriched, and we are not quite sure exactly how, but but we do have some ideas about it. But what we also clearly see, this cluster 7 TAM1, again a TAM1 population, they certainly seem to be coming from these monocytes that are getting recruited into the tumor microenvironment, and suggesting that there's probably these monocytes that are recruited into the TME, they somehow get reprogrammed into these TAM1-like populations, right? In this one, these arrows basically are not coming from any of these other clusters, suggesting that these are tissue residents, right? And I'm gonna come back a little bit later as to the mechanism here in terms of the RNA velocity, but the main take-home message from this is that it appears that, you know, you have recruitment of monocytes into the tumor microenvironment that then somehow get reprogrammed into these TAM1-like population. So then the question obviously is that, well, how do these TAM1 macrophages acquire this particular cell fate, right? And of course, what are the molecular regulators of these TAM1 fate? So in order to look at that, some of the hints actually came from looking at endothelial cells, right? So switching gears just for a little bit, right? So some of the insights that we were looking for as to what's reprogramming these myeloid cells appear to be coming from actually a closer look at the endothelial cells. So again, like I mentioned at the very beginning, in that overall macro clusters, there were endothelial cells that were also spatially distributed between what was coming from normal and what was coming from the tumor. And you can see this again in yellow, specific, these clusters 2, 5, and 6, mainly coming from normal, tumor adjacent normal, and all of these clusters coming from either the tumor core or periphery, right? Now, amongst these clusters, what we were quite fascinated by is this particular cluster, all right? These clusters of 9, 3, and 4 that appear to be expressing very similar markers, and one of the markers turns out to be this gene called PLVAP, all right? Again, without getting into too much details, but this is a plasma lemma vesicle associated protein. It has been shown to be upregulated in a variety of solid cancers, in fact, in the endothelial cells, and it appears to be regulating, or it's thought to be regulating the permeability of endothelial vessels, all right? So especially important in terms of intravasation, extravasation, or as, say, immune cells come into the tumor microenvironment through the capillary system, how they then move into the tumor stroma and the microenvironment. So it's interesting to see that, you know, we find these particular endothelial cells. What's more interesting is that we almost never find them, the expression is very low or almost absent in the normal, right? So we only find them in the tumor, again, not in the normal, right? So then the question is, of course, what's so special about these tumor-specific PLVAP positive endothelial cells? Sorry, I just forgot to mention, we also found another cell type, which is an these are HLA-DR positive as well as PLVAP positive. So we found an antigen presenting endothelial cells. And, these days, of course, in the single cell papers, there are all kinds of antigen presenting cells apart from the dendritic cells, which are the professional APCs. But, CAFs can be antigen presenting. And what we find is that, some of these endothelial cells could also serve as APCs. Sorry, go ahead. Sorry, was there a question? Okay. I guess not. So the question is that what's so special about these tumor-specific endothelial cells? And this is where we started working with Florent Genoux, really because, well, firstly, we found this really interesting tumor-associated macrophage population, and Florent is all about myeloids, of course. And as he's shown that these human fetal dendritic cells actually promote this immune suppression through arginase 2. But we looked at some of these other papers, and there was a 2016 paper in Nature which showed that, and this is not in human, by the way, this is in mouse. So in murine liver, what it showed that PLVAP positive cells in fetal liver endothelium is absolutely essential for the seeding of tissue resident macrophages, right? And that started kind of, got us thinking that this could be really exciting, that maybe that you have these, tumor specific endothelial cells that are actually never found in, at least in the adult normal, but it's possible that it's actually found in, at least in this case, in fetal liver in the mouse. But so it's possible that there is fetal-like, reprogramming of the endothelial cells in the tumor microenvironment. And then these PLVAP positive cells, as has been shown in the mice, perhaps even in HCC, then they start recruiting the myeloid cells, so monocytes in this case, and then reprogramming them into TAM1-like macrophages, all right? So that was the hypothesis that we wanted to test. So before getting into that, so that's just, what I talked about, that they're found in fetal liver organogenesis, and they regulate the seeding of TRMs. So before getting into that, we wanted to make sure that indeed that this is true, that what we find in terms of these endothelial cells are in fact more fetal-like. So the endothelial cells are going back in time to becoming more fetal-like, or what you would see in fetal liver. And in this case, we collaborated with Jerry Chan at the KKH hospital. And so we looked at four fetal livers, 14 weeks, 16 weeks, 18 and 22 weeks. And then we basically went through the same 10x pipeline. And now what you see over here is basically a UMAP clustered and integrated together with the tumor, all right? So the first UMAP that you had seen of the macro clusters was basically only tumors and adjacent normal. In this case, now we've integrated with the fetal liver, right? And what you see is this majority of this orange and some of this blue. So all of these clusters on the left, they're coming from fetal liver. Everything else is basically tumor, all right? And again, how we identify the different cell types and subtypes, I'm not going to go through it again, but basically, find the same immune cells, stromal cells, and epithelial cells. Now, how do we, so going back to the question, right, the tumor-associated endothelial cells, which are expressing PLVAP, are they similar or different from the fetal liver endothelial cells? And now this is basically subclustering just the endothelial cells obtained from this fetal liver 16 and 21 weeks together with adjacent normal and tumor. And so what you find is, okay, you find these clusters, right? This is coming from normal. These are coming from tumor. However, and these are mainly all fetal. However, these clusters that you see on the top, which is surrounded by this red outline, they're coming both from tumor as well as fetus, right? And as it turns out, these fetal-like, these cells, right? These endothelial cells are the ones that are expressing PLVAP. So it's expressed in the tumor cells as well as in the fetal cells, right? And of course, you find a whole bunch of other markers which are again also tumor-specific endothelial cells, which I'm not going to go into it for today's talk. But what's interesting is that there are there is a subpopulation of fetal-like endothelial cells that in fact do cluster or transcriptomically extremely similar to the tumor epithelial cells, suggesting that the fetal liver gene expression program, that there is a fetal-like gene expression program in the tumor endothelial cells, perhaps by some kind of a reprogramming, right? Before I get to that as to how to endothelial cells got there, let's go back to TAMs, right? So we started off asking this question, well, how do we get this TAM1 reprogramming, right? So what we find is that when we start reclustering the myeloid cells, now together with the fetal liver, so again 16 weeks, 21 weeks, adjacent, normal, and tumor, and this is how the clustering looks like. And what you find, again, much of the normals is on the left-hand side, and on the right-hand side, these two clusters, right? This cluster is pretty much coming only from the tumor, and this one is coming mainly from the fetus, right? From the fetal liver. And what you find again is this particular expression of this gene, right, FOLA2 positive, it's coming mainly from this TAM1 population. So this is the cluster 3 and 9 on the kind of the east corner of this population. So these are the ones that are FOLA2 positive. And again, very closely clustering with this particular cluster, cluster 6, which is coming from fetal liver macrophages, suggesting that there is a fetal gene expression program in these TAM1 populations, right? So on the top of it is the TAM2, which is SPP1. So this particular blue cluster, the red cluster and this light blue cluster is a TAM1 like cluster, again, very expressing markers that are very similar that we showed, that I showed you earlier. So in this case, FOLA2. So again, suggesting that not only do you have fetal-like reprogramming of the endothelial cells, but you have some kind of a fetal-like reprogramming of the TAM1 population as well, right? And again, going back to the papers, the Nature paper that I talked about, right? So there are these PLVAP positive endothelial cells in the fetal liver that are absolutely essential for reprogramming and seeding of tissue resident macrophages, right? And so we started thinking that maybe these PLVAP endothelial cells are then recruiting these monocytes that I showed you in the RNA velocity into the tumor microenvironment, and then reprogramming them into these TAM1 like tumor associated macrophages, right? So before going into that, like, is that true or not, we also started looking to see that is it a phenotype that we see in terms of this kind of almost this oncofetal reprogramming that we call it, in this case of the tumor microenvironment. Is it specific to just tumor, or is it also involved in other liver disease? So in this case, what we've done is we've generated some of our own data, but as well as downloaded some of Neil Henderson's data, who works on NASH, NAFLD, and cirrhosis. So there's a paper that came out, I think it was in Nature in 2019, on cirrhosis. And then we integrated the data on cirrhosis together with all the data that we've generated for fetal, normal, adjacent, normal, and tumor. And look to see how it looks like in terms of endothelial cells or in terms of the tumor-associated macrophages. Now, again, what you're looking at here, right, is, so these are all the different clusters. These are only endothelial cells clustered, okay? Together with, again, you're looking at healthy normal, adjacent normal, cirrhosis now, tumor and fetal. And you see this PLVAP expression in two different clusters, right? And the main difference is that these are mainly PLVAP positive, and these are also the PLVAP positive, but also antigen presenting. They have these HLA-DR, as well as a whole bunch of HLA antigen presentation machinery. But this particular cluster on the right-hand side is coming both from really the tumor, which are the red dots, as well as cirrhotic liver and the fetus, all right? Fetal liver, suggesting that perhaps this kind of reprogramming is really an outcome of kind of a wound healing or a chronic wound healing program in the liver, where we look at, in fact, and now we've looked at NASH and NAFLD as well, and we find that as well as partial hepatectomy models, which I'm not going to go into in the interest of time, but we find that in liver injury models, basically, and in cirrhotic models, you find the same reprogramming of the endothelial cells, and not just of the endothelial cells, but also that of the macrophages again. So if you take just the macrophages from all these different time points, right? And combine it with cirrhotic liver and tumor, again, you find this particular TAM1 cluster right here, which is again getting contribution from tumor, cirrhosis, and fetus and fetal liver. Okay, suggesting that it's really that this fetal-like reprogramming that we see of the endothelial cells in the macrophages could simply be a wound healing response, right? Okay, so coming back to well, how do we get these cells, right? These TAMs getting reprogrammed, and is it because of these tumor endothelial cells, which are fetal-like tumor endothelial cells that somehow recruit these monocytes during neovasculogenesis and then reprogram them into macrophages? So in order to look at that, what we first did was to look at what are the downstream pathways that are activated, right? And in order to do that, what you can run is something called CNIC. And this was, again, an algorithm that was developed by Steinhart's group. Um, um, and what CNIC does is basically use a single cell transcriptomic data and then cluster cells based on common regulons that are active, all right? So in this case, you're not clustering cells based on transcriptomic similarity, but you're looking at regulons, multiple regulons that are active, and you cluster cells with shared regulons, right? And the way the regulons are looked at is basically they look at co-expression of genes that are co-expressed with certain transcription factors. And then in those co-expressed genes, they look at their enhancers and the promoters and look for binding sites, predicted binding sites of those transcription factors. And so if you have co-expressed genes with transcription factors for which you have bona fide binding sites, then they consider that as a regulon, right? And then they look at multiple regulons that may be active in a given cell type, and then you're clustering cells based on similarities of regulons that are active. All right, so this is again a Louvain-based clustering. There are different clustering mechanisms, but this is what you're looking at, just simply clusters. This is basically what you're looking at. Well, let's look at the right-hand side, the different clusters where they're coming from. Again, red is a tumor, yellow is a normal, and then this dark and light blue or purples is fetal liver from 16 to 21 weeks. And again, you could see that this particular cluster in the middle, right, these are the cell identities, right? These, and again, we've only done this clustering based on the tumor-associated macrophage, on the myeloid cells, all right? Not just the TAMs, obviously. So these are the monocytes, and these are the TAMs. And you find this cluster on the right-hand side, right, which appears to be, again, FOLA2 positive, and they are like also has these fetal-like tumor-associated macrophages. And one of the top regulon that appears to be active is HES1. And for those of you that know some of these signaling networks, I'm sure you probably immediately know that HES1 is downstream regulator or downstream target of Notch activity. And so what it suggests that there is Notch signaling that is active in these tumor-associated macrophages, specifically these TAM1 positive, or TAM1, the FOLA2 positive TAM1 states. And it appears to be, it's possible that, the activation of Notch signaling is driving this TAM1 cell state. Of course, there are different regulons that are active in different TAM populations as well as monocytes, which are very distinct from this HES1 regulon, which appears to be highly upregulated in the FOLA2 positive TAM1 population, as well as the fetal liver macrophages. So what I'm showing you here is again further proof that indeed there is activation of Notch signaling in the TAM1 population. If you remember, these are the clusters based on fetal liver, adjacent normal, and tumor, and you can see that these are the cells that are expressing CD163, 206, again those immunosuppressive markers, and these are the same cells that are co-expressing HES1, right? So these are the same cells. This is again coming from the fetus. This is the TAM1 population. They're expressing the same markers as well as expressing, now in this case, it is just a transcription readout, right? So expression of HES1 rather than the regulon. And so just to, just to zoom in, it seems like the HES1 expression is actually very specifically there in the TAM1 population, as well as some of the fetal liver macrophages. So, well, if Notch is getting activated, where is the ligand coming from, right? And to cut a long story short, as it turns out, the ligand basically for Notch2 is basically coming from these fetal-like endothelial cells. So remember these PLVAP positive cells? These are the same cells that are actually also expressing high levels of delta 4. Of course, there are other endothelial cells, which is this IGFBP3, which is another tumor endothelial cells, which also seems to be expressing some degree of delta 4 expression, but vast majority of delta 4 expression is coming from these the tumor associated endothelial cells. And also this little cluster over there is the one that was coming from the fetal liver endothelial cells. All right. So now it suggests that, maybe delta 4 expressing or PLVAP positive endothelial cells in the tumor that have been reprogrammed into these fetal-like endothelial cells that are expressing delta 4, it appears that they could be then recruiting the monocytes, as is evidenced by RNA velocity, and the expression of Notch2 is well known in monocytes. And when they come into this tumor microenvironment, then there is delta 4 Notch expression, and which then activates this regulon, which is Notch activity in terms of HES1, so activation Notch, and that most likely is driving this TAM1 fate, right? So can we show this using functional assays? So in order to do these functional assays, what we did is that we got these delta 4 expressing delta 4 coated beads, and then co-cultured these delta 4 beads with monocytes that we isolate from the tumor adjacent to normal, right? So we want to recreate the same scenario in an in vitro test where we take the tumors, right? We go back to the same patient's tumors that we had leftover tumor material from. We take the adjacent normal, we isolated the monocytes, which we think are otherwise recruited during neovasculogenesis and then reprogrammed, right? So we isolate the monocytes from the tumor adjacent normal and then co-culture it with the delta 4 beads, and then simply look at CD163 and 206 as well as FOLR2 expression, which I thought I had, which disappeared for some reason. But anyway, you have to take my word for it. So what you see, this is a control, so 2.76% in this case that are 163, 206 positive with the nude beads. But when we co-culture with delta 4, you see upregulation, about a 6-fold upregulation or a 5-fold upregulation of 163-206. So that, again, that TAM1 signature. And now, which can then be blocked with anti-delta 4 antibody. So, and this is quantified over here, suggesting that it is indeed this delta 4 Notch2 signaling from these fetal-like endothelial cells that may be recruiting and interacting with the monocytes expressing Notch2 and then reprogramming them into TAM1-like cell states or cell fates by the activation of Notch signaling. And what we've now looked at is that we've now compared these regulons that are active, right, across what we find in the TAM1s of the fetal-like macrophages, as well as embryonically seeded murine liver macrophages. So I'm not showing you all the data on how we generated this per se, but we have these mice from Florent Genoud's lab, which are marked with specific TD tomato, and you can look at the ontogeny based on whether this TD tomato is expressed or not, and you know which one is embryonically seeded versus which one is monocyte derived, right? And so what's really interesting is that when we look at all these regulons, right, these regulons seem to be this particular module 2 of this regulon seems to be highly active for Notch signaling, has HES1, of course has some of these other regulons that are also active. But what's important is that we find this regulon active not just in fetal liver and tumor associated macrophages, the TAM1 population, but also in the mouse embryonically seeded macrophages, which are again known to be monocyte derived, all right? So suggesting that in fact, it is evolutionarily conserved, right? So these same mechanisms and regulatory activities or regulatory signaling pathways that are involved in seeding the liver resident macrophages are the same ones that are present in the human fetal liver, and the tumor-associated macrophages that are getting reprogrammed by these fetal-like endothelial cells in these HCCs are basically using the same kind of processes to drive this TAM1-like phenotype. All right. Okay, so that's what's happening with TAM. So now if you go one step upstream, you could ask, well, where are these tumor endothelial cells coming from? So again, we ran RNA velocity, and RNA velocity is suggesting that this is all happening, all the tumor-associated fetal-like or embryonic-like PLVAP positive endothelial cells are coming into the tumor through neovascularization. I'm not going to belabor this point so much. But what's important is to understand is what's specifying this tumor-like or tumor endothelial cell fate, or this fetal-like endothelial cell fate. So in this case, so I've already talked about cell phone DB, but there is another algorithm that you can run. It's called NicheNet. And what it does, it not only does it look at ligand and receptor expressing cells, but it'll also look at downstream target genes in the receptor expressing cells. So it'll make these or infer these connections between two different cell types based on the ligand that is present, the receptor, as well as the activation of downstream target genes. So in some ways, you have a lot more confidence of these inferred interactions being true, because not only do you have the expression of the the ligand receptor pair, but you also have the activation of the downstream regulatory network. And again, cutting a long story short, what we find is that most likely, and this is not necessarily that surprising, VEGF signaling, all right? So tumor hepatocytes, and this is actually quite interesting, tumor hepatocytes have expressed high levels of VEGF. And what's in fact known from a previous paper, that some of the downstream target genes of VEGF happens to be PLVAP. Now, endothelial cells, of course, express very high levels of VEGF-R2, and in fact, it is especially high in these PLVAP positive endothelial cells. So you can see these are the violent plots, right? So this is PLVAP positive, and these are the same clusters, the cluster 3 and cluster 4, that the PLVAP positive cells are expressing higher levels of VEGF-R2. As well as there's another gene called NRP1, and we can discuss this later. I mean, there are interesting biology there that I won't have time to talk about. But what NicheNet suggests is that you have the hepatocytes that are expressing VEGF from the single cell data. And then of course you have PLVAP expression of VEGF-R2 in the endothelial cells. So what's most likely is happening, so the other interesting thing is that in the normal, adjacent normal, in a non-dividing hepatocyte, you actually never see VEGF expression, all right? So it's only dividing, actively proliferating hepatocytes that are secreting VEGF, which then, which happens in the tumor, obviously, and you can imagine it happens during partial hepatectomy and regeneration, and in cirrhotic liver, or any kind of wound healing, kind of tissue repair kind of a model, where you have actively proliferating hepatocytes which are now expressing VEGF, which then interact with these PLVAP positive, which interact with the VEGF-R2, which is expressed in the endothelial cells, and activate this embryonic or fetal-like program in endothelial cells, which of course then go on to express delta-4 and recruit monocytes and reprogram them into the TAM1 population, all right? So that's kind of, the hypothesis that we're working on, and hopefully, I've shown you a variety of different ways of showing that, but of validating that. But of course, the proof of the pudding is always in being able to visualize some of these interactions. And so we used clinical validations using spatial transcriptomics. So what you're looking at over here is a nanostring panel that we've looked at. So we generated a customized panel of about 96 probes. So these are RNA probes. So you're doing 96 in situs all at the same time on FFPE sections, all right? And these probes are basically conjugated to oligo barcode or oligonucleotide barcodes through photocleavable anchors. And what happens is that, so you would just simply stain, in this case, so what you see is a staining, as a 4-color staining of DAPI. Actually, let me just show you here. Yeah, so This is the digital spatial profiling platform, or DSP. So you color them with, say, a cytokeratin epithelial marker, immune marker CD45, CD34, as well as DAPI, and then you look at these regions of interest, and these round circles are basically specified by us. So this is on the left hand side is normal, on the right hand side is a tumor, right? And now if you zoom in, this is how it looks like, right? So now in these regions that you have highlighted, the regions of interest, you can use this nanostring GeoMX. What it does is goes and shines UV light, and the cross-linking is basically, it's uncoupled, and these oligonucleotides, which were otherwise hybridized to the probe in these different areas of the tissue sections, the tumor section, it gets sucked up into the system and goes into a 96 well plate, and then they use encounter or sequencing based technology to simply quantify the levels of expression of certain genes. So in this case, you're quantifying the levels of expression of 96 genes at the same time in 12 different regions of interest in the normal and the tumor. And we did it not just in normal and tumor, we also looked at in fetal liver, adjacent normal, and tumor, all right? And that's what we basically trying to look at. So, you know, all this while I've been talking about a tumor environment and an ecosystem. So in this case, the regions of interest are basically selected ecosystems that we've chosen within the tumor, the tumor periphery, and outside the tumor in the normal and stroma, and trying to see, well, what kind of expression of these genes that we've identified with single cell analysis do we identify in terms of spatial organization of these genes. And what was immediately evident to us is that, and this is quantification of the data, so it's very, very sensitive and highly quantitative, is that we get these two major clusters, right? All the regions that we find, so these are now about 108 different regions across 6 slides, so 2 fetal, 2 normal, and two tumors. Yeah, and so we find that there are 2 major clusters that we find, and you can see this cluster 1, which is basically this cluster on the left. It's mainly getting contribution from fetal tissue, which is this light mauve over here. And then the red is tumor, much less or very little from adjacent tumor normal. And cluster 2 appears to be mainly coming from the normal. And of course, there is always some contribution from the tumor and the fetal fetal tissue. But what's really evident is that when we quantify it, right, cluster 1, which is mainly coming again from the tumor and the fetal liver, it appears to have all high levels of expression of all these oncofetal markers. So high levels of FOLR2 positive TAM1s, high levels of PLVA positive endothelial cells, there's high levels of Notch, high levels of VEGF signaling, high levels of FOXP3, CTLA4, regulatory T cells, BATF, TIGIT, LAG3. So you could see that all of these genes, which are very highly expressed, right, they're all in this cluster 1 per se, and these are all representative of an immunosuppressed oncofetal-like ecosystem. However, if you look at cluster 2, which is adjacent normal, they have basically the opposite expression of many of these, you know, the genes that we talked about. So validating this suggestion, this hypothesis that, you know, in the tumor, in HCC at least, you know, it's undergoing kind of an oncofetal reprogramming of the tumor microenvironment, which then helps the tumor cells to survive and grow, right? So that's then comes to, you know, bring me to the working model that we have. So again, like I said, you have these normal hepatocytes, and this is my last minute effort on just drawing it on my iPad. But so, you know, in red is the hepatocytes, and then you have, you know, the immune cells and the fibroblasts and endothelial cells. Now in the tumor microenvironment, you know, you have, of course, you know, you have some kind of an insult, there's mutations and environmental insults. And so these hepatocytes start proliferating. Now, as they proliferate, like I said, these proliferating hepatocytes are the ones that are making VEGF, right? And this VEGF would then interact with the VEGF-R expressing endothelial cells and activating this VEGF signaling in these endothelial cells, which then generates these embryonic or fetal-like PLVA positive endothelial cells. Some of them will be expressing delta 4, And during neovasculogenesis, like I showed you with RNA velocity, both for the endothelial cells as well as the TAMs per se, that these endothelial cells, when they come into contact with these macrophages, or the monocytes, sorry, which are expressing Notch2, then they activate the Notch signaling, and what we see is with HES1, Regulon basically gets activated, and then they get reprogrammed into this TAM1 like populations that are FOLA2 positive, CD163, 206 high. They express PDL1, CD86, many of these immunosuppressive or checkpoint blockade ligands, which then interact with CD8s and CD8 and NKs perhaps, and perhaps result in immune suppression. But what's also interesting is that they also start recruiting the regulatory T cells, which are the FOXP3 positive Tregs, and recreate this immune microenvironment, which is highly immunosuppressed, all right? And because of this immunosuppressed microenvironment or immune tolerant ecosystem that this creates, these immune cells can no longer basically target the hepatocytes. So in some ways, what we think is what the hepatocytes are doing is that it's not only do they proliferate, but they reprogram, or they start this cascade of reprogramming event to make the ecosystem beneficial for its own survival and growth, right? So firstly through the endothelial cells, going into the immunosuppressive tumor-associated macrophages, which then would have its final effect on the T cells. And so this is a prettier picture, of course. Just to summarize what I've talked about, and I think what's really interesting is that we find this not just in the tumor microenvironment, but also in the environment of chronic liver disease such as cirrhosis, as well as a partial hepatectomy model, which I didn't have time to show you here, but it's there in the paper. The paper is going to come out sometime mid-October, so you can read about it then. But what's really interesting is that in partial hepatectomy model in the mouse liver, we can show that when we took it day 0, day 7, day 14, day 21, so by 21 days, the regeneration is complete. But by day 7, all of these oncofetal reprogramming markers come up, right? PLVAP, FOLA2, all of this. So there's this signature of oncofetal ecosystem that comes up early on during regeneration, which then dies away as regeneration is complete, right? Suggesting that this kind of a wound healing process, and if you think of as cancer as a non-healing wound, then it kind of makes actually perfect sense that they basically seem to be going back in time, back to basics in some ways, in terms of how they developed during liver organogenesis, and using those same mechanisms to create an immunotolerant environment which allows the growth and survival of these hepatocytes. And finally, a broader conclusion in some ways that we think that single cell RNA-seq, not only does it allow us to dissect intrinsic and intrinsic factor that guide evolution, what it's telling us that maybe as we do or as we identify or make such discoveries, that maybe we could start thinking of targeting the tumor microenvironment rather than the tumor cells directly as a therapeutic intervention strategy in liver cancer. And clearly there are obvious consequences for immunotherapy as well as novel biomarkers for patient stratification. And this is particularly true in the light of this new study that was just, published just 2 months ago, I think, in New England Journal of Medicine. It's called the I Am Brave 150 trial. I think it's written somewhere over here. But what they looked at is about 500 patients with locally advanced hepatocellular carcinomas. They were all metastatic, unresectable, or both, right? And where they found that compared to sorafenib, which you might know works in maybe like 5 to 15% of HCC, it's really a bad drug actually. Well, there's nothing really that's good for HCC that in terms of drugs that work, but which is why there's a lot of interest in immunotherapy. But one of the combinations that's working or apparently working really well is the PD-1 inhibitor. This is the Roche drug. It is a lizumab, which is anti-PD-1, together with Avastin. Bevacizumab is basically the anti-VEGF inhibitor, right? And that seemed to have given quite a remarkable response, clinical response in HCC. And we think that we have the molecular basis to explain why that would work well, right? Again, we think anti-VEGF, right, would probably prevent this reprogramming of this fetal-like endothelial cells, which will perhaps then reduce the generation and reprogramming of the TAM1-like populations that are immunosuppressive, and therefore they'll recreate an immune or inflamed microenvironment where the immune cells can come in, cytotoxic T cells can come in. And then, of course, in the presence of PD-1, it can really kind of have a synergistic effect on having a better treatment outcome. So we think we can stratify patients, basically who's going to slated for immunotherapy, for who may or may not get this combination with bevacizumab with anti-PD-1. And finally, like, this is more of a philosophical discussion. I mean, I find it absolutely fascinating that, you know, cancers seem to be echoing the embryonic past. And this is not something absolutely new. I mean, if you look at the literature, right, oncofetal genes, right, have been identified, or even the notion of cancer stem cells, that they become poorly differentiated or undifferentiated, they're going back in time, right? The cancer stem cell hypothesis. So that's all, of course, you know, well known, although there's still a lot of controversy. But what we are seeing here is that, in fact, the tumor microenvironment can also go back in time, right? And the reason it's probably going back in time is that, in fact, you find these amazing, remarkable similarities between, you know, the fetal-maternal interface between the placenta and the developing embryo and the tumor, right? And that's shown over here. But one thing that we know, developing fetus, right, a growing embryo and developing fetus during organogenesis is extremely immunotolerant and immunoprivileged, because otherwise you'll get rejection. And mainly because if you look at a growing embryo or a fetus, I mean, it's basically full of all the hallmarks of, you know, the classical hallmarks of cancer. There's migration, invasion, intravasation, extravasation, you have cell shape changes, you have tremendous amount of proliferation, right? And, you know, classical example, gastrulation itself, they in fact do exhibit several of these hallmarks of cancer. So, you know, the way we are thinking about it is that, you know, the reason it perhaps, you know, this reprogramming is required is really to kind of go back in time, so that to recreate that same kind of fetal-maternal interface in this case, or the same fetal-like environment, which really promotes the survival of these malignant cells, right? So the questions that, you know, we're very interested in is that, you know, how do these cells recall this memory of this past? Is something to do with epigenetic memory that is getting reignited? What are the conserved developmental pathways? Of course, you know, Notch being one of them, and VEGF, and how perhaps mutations in environmental insults then influence this memory and epigenetic regulation. All right. So, okay, I'll just end with that. Let me just acknowledge. I think I already acknowledged Ankur, who started the liver project and some of the single cell projects. But what I didn't have time to talk about is some of the work that's happening now in NPC and nasopharyngeal cancers. In fact, we are looking at pre-post immunotherapy plus minus bevacizumab with endothelial, with a VEGF block. Jamin is working on using single cell, again, transdermics to look at metastatic colonization of colorectal cancers in the liver. And of course, you know, many of the, you know, the lab superheroes, like, I'm just on the sideline, you know, the cheerleader, really encouraging them. But there's some phenomenal work being done by these people. And of course, you know, patients, we've been extremely privileged and very grateful to the patients and their family who decided to contribute the samples for research. And of course, all the clinicians for a variety of different cancer types, especially peers for HCC for the story, but a whole lot of other clinicians that we're working with for colorectal and head and neck and breast cancer, who've been amazing at providing these samples and the clinical insights into this data. All right, so with that, I'll thank you all for listening. I'm sorry I probably went really over time, but and happy to take questions.
Fantastic job, Ram. Thank you very much. Super inspiring. I think we are all applauding. Yeah, applauses are coming up. Great, thank you. Yes, indeed, we lost a few people during the hour marker because a lot of students they have classes, so.
No problem at all. Yeah.
Okay, thank you very much. So maybe I start with the first question,
Yeah.
which is more of a general one. So you have shown, and I was fascinated by the fact that you can also trace in time the genetic profiling or the transcriptomic profiling. But I wanted to ask you how this relates with the tumor progression. So do you have also samples of tumors at different stages that
Yeah.
you can analyze?
So these were all, so we do not, so most of the tumors that they were resected were fairly advanced tumors. We do not have very early ones, but like I said, we've looked at cirrhosis and NASH-NAFLD, so early liver injury models, where we do see the same program getting upregulated. One thing that we have amongst these 14 patients, there are 7 that have relapsed. And we have done the single cell RNA-seq on it, but it's getting analyzed right now. So in that case, from the same patients, basically over time now we'll be able to tell how the tumor microenvironment as well as the tumor cells may have changed during recurrence or relapse, but we don't
Fantastic.
have the data. Yeah.
No, it's fantastic. I feel you're sitting on a mine of data here. There is stuff to dig for years, probably. Okay, so questions from the audience?
I have a question, but I don't know if you can hear me okay, because I'm currently in the car traveling south, but
We can hear you.
yeah, I had a question in regards with what you talked about, the embryo process. Would you expect in the future, like if you have a long period of a cancer development, would you expect it to show more and more differentiation inside the tumor, maybe some new regions, and just like more polarization of yeah, the cells just having different poles with starting to develop some kind of different functions, kind of like in an embryo?
Right. I mean, I think that's an interesting question. I think, well, from a tumor perspective, right, when you have the mutations and from a tumor intrinsic property, many of them do remain undifferentiated, right? So we find, yeah, no, I mean, so we don't really see, okay, let me just say, so for HCC, we've looked at some specialized differentiation markers, and those differentiation markers are generally expressed at low levels because they remain more malignant, right? So, and they keep secreting VEGF in this case. So the difference between cancer and say regeneration, we see this uptick, but it's temporary, of this oncofetal reprogramming, and then they stop, right? Because reprogram is complete and then they differentiate. But I feel that at least in HCC, as long as it keeps dividing, you don't see as much differentiation happening, all right? And that's purely because the malignant cells have these inherent mutations, and they appear to remain more poorly differentiated. Yeah.
Thank you so much, and wonderful presentation.
Thank you.
I have a question. Thanks for the talk. It was an incredible amount of data. I have basically like a methodology question first. That of the 14 patients you took initially, were those patients all in stage 3, 4 cancer?
Yes.
Did they have metastasis?
No, these are
No?
all stage 3, 4 because metastasis we cannot resect.
Yeah, okay, but they didn't have any known metastasis because it's and
That is
for
correct.
example, yeah, and did they? I mean, did you control for any pre-existing conditions or the amount of if they had any fatty liver
So
before?
exactly. So there are definitely some that were cirrhotic, right? They had cirrhosis, and the only other thing is, like I said, they have Hep B positive and negative. some patients that are Hep B positive and negative. And if we cluster, so we didn't find any, say, Hep B positive negative clusters that are separate, right? So they are not segregating into different clusters based on other pre-existing conditions that at least we could identify. Certainly not the tumor microenvironmental cells, right? They were segregating based on normal and tumor, but not other pre-existing conditions. But it's something that probably we need to go back and take a deeper look, but at least from the overall view, we don't really see any specific effect. But to answer your question further, like I said, so we have now about 5 patients who have relapsed. And in that case, they also have like locally advanced metastasis as well as distant metastasis. And so we're getting biopsy samples. And in those cases, we'll be able to tell, at least try to kind of, probe more into what's happening to not just the tumor cells, but also the microenvironment. Yeah.
It would be pretty interesting to look at the same profile that you did, but after some specific therapy, after chemotherapy, after
yes.
Yeah.
So we are working with Merck actually. So MSD, Merck over here in Singapore, they're running an HCC trial on exactly that. So they're running a trial on their pembrolizumab, which is an anti-PD-1, and plus minus a 2-arm study, plus minus bevacizumab, which is the anti-VEGF. So if it works, well, of course, you hope that it works on patients. But the issue always is with clinical samples, when it works really well, you don't really get a sample post-treatment. But in the patients that don't respond, we'll be able to then hopefully identify new biomarkers, or whether in the tumors or in the microenvironment, that may be prognostic of resistance. But we can always look at the pre-treated samples basically in that trial setting, to look to see who responded better and why. But like I said, this study itself is telling us, in light of the I Am Brave clinical trial that I talked about in the NEJM paper, that most likely the patients who have this PLVAP positive endothelial cells, they are the ones that we think would most likely respond the best to combination combination immune checkpoint blockade. Sorry. Yeah.
Excellent. And I think you're looking for other immune checkpoint receptors also.
That is correct. That is correct. Not
CD96 and
yeah, exactly. Yeah.
Cool.
I have to say, with immune cells, sometimes, so single cells have a major caveat of single cells, that there's a lot of dropout events as well. So you can see it at the level of protein, but at the level of RNA, you simply don't capture it sometimes. So you cannot capture everything, but whatever you can, you just try to make it up by sequencing enough number of cells so you can, capture something. But there's not always a one-to-one correlation between the transcriptome and the proteome specifically for immune cells, because, just having the transcript is not enough, which is why we always try to validate with flow as well, because the cell surface localization becomes really important. Yeah.
Thank you very much.
Sure.
Great. So I would have another question for you, which is about the reprogrammed cells and their origin. You said that comes from neovasculogenesis. My first guess, it was that it could have been the tumor stem cells or the bipotent resident cells. So what is the evidence for the neovasculogenesis as origin?
You mean like that, what's it called? The vascular mimicry?
Yeah, exactly.
Okay, so
the transdifferentiator.
So exactly well, possible then, but I would think that they would be expressing at least some markers of epithelial cells. We don't see that. And secondly, like when we do RNA velocity, we do not see. So RNA velocity takes all genes, all transcripts, and we do not see any cell. So, what I showed you was just with endothelial cells, but of course we've run the RNA velocity, say, taking into all cells into account. Okay. And we don't see any arrows coming from the epithelial compartment into that into the PLV positive endothelial cells, suggesting that most likely it's not. But it's very hard to completely rule that out. But that's something that I've always been fascinated with. But we're just, but I think we don't have. So see, this is where you need a really good mouse model. With human samples, you have what you have, and that's what you sequence, and that's where you would love to basically mark those cells. And if there's a good HCC model, that too immune competent, right? HCC model, which there isn't. I mean, the HCC models are really, there aren't any good HCC models actually. But if there is, and you not need an immune competent model, but then you can mark those cells, right? The epithelial cells, and then see if they now become they acquire fates of endothelial cells. Yeah, would be fascinating. Yeah.
Right.
But at least with RNA velocity and at least with clustering, it's a very, very different cell type, and there is almost no similarities. That's why we think it's not mimicry. Yeah.
Or even more simple, you could make a PDX right inside the liver. I don't know if that is possible, but with human cells, and then see if the reprogramming comes from mouse cells.
Right. Yeah, that's a good idea actually. Yeah, we could definitely try that actually. Thanks. Yeah. That would be very interesting to look at. Yeah. It's just that only thing is with the PDX models, I'm always a bit apprehensive because I feel like, if it's not an immune competent model, I mean, you can look at just that particular aspect, of course, that's a different thing, but I don't know what else in the immune system is also promoting this kind of plasticity. And what if you don't see it, doesn't mean it's not there, or you just, don't have the right model. So
you definitely miss out a lot of the complexity. Yeah.
That's right. Yeah. And again, but that said, the mouse immune system is actually fairly different from the human immune system as well. So you really need kind of a humanized liver grown in a humanized immune system.
Right.
Yeah, but then the cost also becomes prohibitively expensive. So we are trying actually the same thing in syngenic mouse models. And we'll be able to do that. So like I said, we have these mouse models which have a Cre recombinase driving tomato if it's monocyte derived versus yolk sac derived, right? So we know where the monocytes are coming from. So we know where the macrophages, they're definitely for sure coming from, recruitment of monocytes for sure from the bloodstream, and then they're reprogrammed, right? Because we can mark those cells, we seem to see the same cells in mouse liver as well. And in partial hepatectomy model, we see it is always the monocyte derived macrophages that are giving rise to the STAM1-like signatures. But for the endothelial cells, the thing is we don't really have a good marker. I think we could look at it there, but we don't, again, that's a partial hepatectomy model and we don't have, So anyway, so what we are trying to do is a syngenic mouse model, but we tried that, actually reviewers asked, but then COVID happened and so we injected mouse liver cancer cells into the mouse liver, but the take rate wasn't, there was a lot of rejection because it needs to be HLA matched and things like that. So then there are a lot of, it's not a trivial experiment to do.
Yeah.
But I'll definitely think about, looking into that actually.
Okay, great. Yeah. So are there any other questions?
Oh, yes, please. Yes.
Charlotte.
Yeah, hi. Thank you for your presentation. I really loved it.
Thank you.
I have one question though. I noticed that some of your results you have that between cell interactions, you see a lot of Gal9 and TIM3, and then Gal9 and CD44 when you look at macrophages and other immune cell interactions like CD8s or natural killers or Tregs. So did you notice, I'm working on that pathway specifically, that's why I'm asking. Have you noticed if this SAM1 phenotype, if this population is enriched in Gal9?
So, sorry. So, is a question that whether the TAM1 population is enriched for the LGAL S9?
Yeah, exactly.
Okay. Yes. So those are specifically expressed in the TAM population, but I'm not very sure whether it's a TAM1, TAM2, TAM3.
Okay.
I know that the TAM1s are expressing high levels of LGAL9, and I'm happy to share the data. Actually, it's you can take a look at it, but it seems to be highly enriched in the tumor associated macrophages compared to in normal liver.
Okay.
Yeah.
Okay, great. Yeah, I would
Yeah,
love
it is
to.
something
I would love
quite
to see that.
interesting. Yeah. No, I would actually. Yeah. So, are these the galactins actually?
Yeah,
Do you-
Galaxy 9.
Right. Okay. So great. So this is basically my ignorance. So
No, it's okay.
so which is great because so we are also working on pancreatic ductal adenocarcinomas, and there is a very specific enrichment of GAL9 and GAL3. I think it's actually, it looks really interesting, yes, but we do not really know. I mean, this is where we're looking for collaborations where we have no idea what to do with it, but, or what it means, but it could be really interesting to chat more if you're interested.
Yeah, maybe we can talk a little bit more about it. I've been working, that's my PhD thesis. I'm about to finish at the end of the year, hopefully.
That's awesome.
So yeah, I would love to talk about it.
Sure, that'll be great. Yeah, because we
Okay,
need
Andrea can,
because
Andrea
there are
can send you
things
my
that
email
you
or
see.
something.
Yeah, sure. Because Single said the million things that you see, and really like, million minus one, you don't understand apart from the one thing that you understand. So
yeah, when it comes when it comes to immunology, there is no ignorance because it would be easier to know each star of the universe by name and all the proteins and things that are playing a role there.
I have to say, the joke is all the cancer biologists are becoming cancer immunologists, and all the immunologists are becoming cancer biologists now. But I have to say, like, the last 3 years has been like intensive learning of immunology. But there is so much plasticity in nomenclature, and it's all, you find every marker in every cell type, it's just intermediate, dim, high, bright, low, whatever. But so when you look at the RNA-seq data, it's kind of difficult to know because like I said, when you look at the mRNA transcripts, yeah, you do find it, but when you talk to the immunologists, right, I mean, then they would